In this podcast, we're going to discuss how we can investigate mitosis, which is the form of cell division which produces two identical daughter cells. So one thing that we can use to investigate mitosis is we can prepare squash cells from root tips and look at them under a microscope. So only a few cells are able to continue dividing in a multicellular organism. In plants, the growing tips of roots and shoots contain meristem tissue that can divide by mitosis for growth. So first, you need to prepare the sample. To do this, we recommend wearing gloves and using forceps to handle the tips. The root tips must be sprouting, which means that they're actively growing. And place this into a 5 molar hydrochloric acid solution. And after 5 minutes, rinse the tips in cold water in a watch glass. Then, using a sharp scalpel, cut the root tips that are 2 millimeters long. Place the root tip on a microscope slide and ensure the slide is clean to reduce the chance of artifacts. Then you need to stain it. So carefully add about 2 to 3 drops of the stain and leave it for about 2 minutes. Use a mounted needle to spread out the root tips on a thin layer. And then place a cover slip on top of the tips to protect them. Then apply some force to the cover slip to squash it down. You can either do this with the flat end of a pencil, or the slide could be covered with a paper towel and then you can press it. The force must be vertical or the cover slip might break and move away, or it might injure you in some way. So now you can look at it under the microscope. So place the slide on the microscope stage using the lowest power lens. You then focus the lens on the sample using first the coarse control and then the fine control. So those are the two different dials on the side of the microscope. Then move the slide to see a range of cells. The cells closer to the tip will be those which are more actively dividing. On a lens power of about 400 times, it should be possible to clearly see the chromosomes in the dividing cells, so you can see mitosis in action. And you can work out the Mitotic Index. So this is used to work out the proportion of cells in the sample that are dividing by mitosis. And so the equation for this is, so the Mitotic Index is the number of cells in mitosis divided by the total number of cells. And you can also calculate the size of a cell using a micrometer and a graticule. So the eyepiece graticule is a glass disc in the eyepiece of a microscope. And cells can be seen through a microscope, and a micrometer can be used to calculate the actual size of the cells. In order to calculate the cell size, the eyepiece graticule must be calibrated. The graticule is calibrated by comparing the length of each division in the micrometer to the length of each division in the graticule. Once the eyepiece graticule has been calibrated, the actual cell size can be calculated with an equation. And that equation is the actual size of the cell equals the size of the image divided by the magnification. I'll repeat that for you. Actual size equals size of image divided by magnification. And that's just a brief overview of how you can investigate mitosis.